Bismillahirrahmanirrahim. So let's talk about benign tumors of pancreas. So the first one is serous cystadenoma, which is of completely benign nature. Then there is mucinous cystadenoma and intra-ductal papillary mucinous neoplasm, or also known as IPMN. Serous cystadenoma is a completely benign tumor, whereas mucinous cystadenoma and IPMN have the potential to transform into invasive carcinomas. Let's talk about serous cystadenoma. As the name shows, there is serous fluid, and cyst means a pocket of fluid, and adenoma means benign gland-like proliferation. So serous cystadenoma is completely benign, and it is more common in females than the males, the ratio being 2:1. It is incidentally discovered in 6th or 7th decade of the life. To understand the pathogenesis, we need to know that there is a VHL gene, which is a tumor suppressor gene. So there is a mutation which undergoes a loss of function mutation. So this mutation results in increased and uncontrolled cell growth, forming a benign tumor. So these tumors are most commonly found in the tail of the pancreas, and they have a very small size, which range from 1 to 3 millimeter. They are always in the multiple cysts, as you can see in the microscopic appearance. So under the microscope, they appear as the round cystic lesions filled with the serous fluid. As you can see, the cavities filled with the serous fluid. The inner lining of these cysts is the cuboidal cells, and there are no necrotic areas and no mitotic bodies, which suggest that the tumor is completely benign. Coming on towards the mucinous cystadenoma, again cyst means a pocket of fluid and adenoma means benign gland-like proliferation, which is filled with the mucin. So these tumors are benign but do carry the potential to transform into invasive carcinomas. So 95% of these tumors are found in the females. To understand the pathogenesis, we need to know that there is always a mutation that drives the development of tumor. So there is a mutation in tumor suppressor genes, which are TP53 and RNF43. And there is also mutation in the proto-onco genes, resulting in the uncontrolled cell forming a tumor. So for the exam point of view, you don't need to remember the names of these genes, but you need to remember that there is a mutation in the tumor suppressor gene, which is TP53 and proto-onco genes. So both of these factors result in the increased cell growth, which gives rise to tumor. So almost 77% of these tumors are found within the tail of the pancreas, and they are larger in size than the serous cystadenoma. As you can see the section of mucinous cystadenoma in the diagram. So these cystic cavities in the mucinous cystadenoma contain a thick fluid known as mucin. The inner lining of the cyst is columnar epithelium. As you can see, this is the columnar epithelium. There is also a dense subepithelial stroma. The stroma contains elongated cells known as the spindle cells, as you can see in the diagram. These tumors might contain the invasive carcinoma cells, which can transform into a malignancy. So as the name shows, intra-ductal means within the duct or within the lumen of the duct, and papillary means finger-like projection. So mucinous means mucin producing, and neoplasm means a cancer, either benign or malignant. So these tumors are most commonly found in the males rather than the females, and in 10 to 20% of the cases they are multifocal, which means they are located in multiple places. Coming on towards the pathogenesis, so like all the tumors, there is a mutation in the tumor suppressor genes, which functions as a check against the uncontrolled cell division. And there is an oncogene which normally increases the cell division. So there are loss of function mutations in the tumor suppressor genes and a gain of function mutation in the oncogenes. So both of these mutations result in the uncontrolled cell division, resulting in tumor formation. So these tumors are commonly found in the head of the pancreas and involve main pancreatic duct. So these tumors might also extend down into the minor ducts of the pancreas. So as you can see in the diagram, this is the duct of the pancreas, and this is the finger-like papillary growth pattern that you can see is identified as a neoplasm. So this is the papillary or finger-like growth pattern protruding into the duct of the pancreas. These cells show variable differentiations, which means they range from atypical to the typical cells and have a strong potential to become malignant cells. So there are cysts in the cancers which produce mucin. These tumors act as very strong precursors of the invasive carcinoma. Unlike the mucinous cystadenomas, there is no dense stroma in these tumors. That brings us to the end of benign tumors of pancreas. Thank you.